On the translation. So we are now.
I can be lingual. It's okay. I'm just kidding. Or trilingual. Okay. So we have translators.
No, translation on the YouTube website.
Okay. Okay.
Before you have the conference.
Can we continue? Yeah. Thank you. Okay. So with what I was trying to say, that's a new area. So we live longer, but we would like to have a high quality of life, and I think memory is very important, and we have to be sure that we can do well to the rest of our days. And with that, I think neurodegeneration is the most important area to advance. And I think it's, and I appreciate very welcome to you to this major symposium. I would like to say that National Laboratory Astana is a leading research organization of Nazarbayev University. And I think we have a major and a kind of strong infrastructure. And as you can observe, we have a strong genomics, and I think the best genomics labs in Central Asia that combining most advanced sequencing platforms. And but most important, we have great people, and I appreciate that National Laboratory Astana built a strong research team, research personnel with our administration support. I think we have no excuses, and only one excuse is only right now is a slow procurement, but I think we're still working on it, and I hope we can work it out. So with that, when I say science is very important, it's critical for our development as a nation. So it's one of the critical factor, and I think we have to move forward, and with all our, I think with all our effort. I think with our collaborations today, actually, was very exciting. A lot of European countries, Netherlands, France, I think also Portugal. I think I see the speakers coming for the symposium, for the workshop. Thank you very much. And I think together we can build strong research programs in particularly this important area of neurodegeneration. And thank you very much. That's why I say Koshka lives there. Very welcome. So with that, I would like to also mention that I was very excited to learn about this PhD program from Maastricht University that where actually our researchers can be selected and they can actually do their research here and getting Maastricht PhD diploma from Maastricht. Okay, so I think that's very exciting, and we have a lot of young scientists who actually slow down getting their master, and then they're only doing research. But I think that will facilitate their actually advancement and getting signed a PhD degree. I think we have to work on it, and with that, I'm really welcoming our new research collaboration, but we would like to enhance our collaboration by developing even what's called Hassan, what do we develop? Memorandum. Yeah, that's good. And I would like you to meet now first before Tatyana will start. Hassan, our head of our research office. Hassan can mention more about how can we make a memorandum. Please, just a couple of minutes. Thank you very much.
Thank you. Қайырлы күн, құрметті әріптестер. Сіздерді көргеніме қуаныштымын. So, dear colleagues, so my name is Hassan. So I am, as it was mentioned by Dr. Sarbasov, I am a head of the Research Administration Office of National Laboratory Astana. So mainly we are working with our collaborate. So we are working on the items regarding the collaboration. So we are preparing, drafting the contracts, memorandums with our partners, and trying to collaborate with the different institutes and universities in this regard. So first of all, I would like to welcome the participants of the workshop symposium. It's really great that we have a chance to organize such kind of big, how to say, measure activity that National Laboratory Astana is the part of this big event. And saying very shortly about National Laboratory Astana, I would like to say that we are the main research institute, research organization of the Nazarbayev University. And within the structure of National Laboratory Astana, so there are two centers, Center for Life Sciences and Center for Energy and Advanced Material Sciences. And within these two centers, there are 13 laboratories. And also we have more than 100 research staff under these two centers. So most of our researchers are very, highly qualified. And I would like to say that most of them are internationally recognized. So currently, National Laboratory Astana is conducting two big research programs, and as well as more than 50 research grants under the funding of the Ministry of Science and Higher Education of the Republic of Kazakhstan, and as well as under the funding of Nazarbayev University and other sources of funding. So I would like to say also that we are conducting also commercial projects, commercial grants. In this regard, we are working with the industry, with the business of the Republic of Kazakhstan. And in this field, so I would like to say to our partners that we are open for collaboration. And if you will have, if you will need more information regarding analysis activities, research activities, so commercial grants activities, for example, you can directly contact me or other, for example, research staff of National Laboratory Astana, and we will give more information. So regarding memorandum, this is a practice of Nazarbayev University. If we are, for example, working with the international or local partners, we are usually concluding a memorandum. It's usually we usually conclude this memorandum for 3 years. So in this regard, we have to first of all identify the main directions for collaboration items, and after that, then my office, research office, will contact directly with the institute or university that would like to communicate, to collaborate with the National Laboratory Astana. So colleagues, I will not more take your time anymore. Then I would like to wish you best with the symposium and workshop. Thank you. So thank you very much.
So now it's welcome, Dr. Tatyana Strekalova. Thank you very much. And thank you very much for organizers. I think this was spearheaded by Dr. Askarova, Sholpan Askarova. Thank you very much for the team organizing. Thank you. Appreciate it.
Okay. All right. So hello, everybody. So first of all, I would like to thank this very warm welcome that actually experienced for the first time coming here to Astana and to the university. But now we came with research project, and hopefully it will work out. It's of course an enormous pleasure to find such enthusiastic researchers who are on top of it interested in similar things we are working on, such as problems of neurodegeneration, stem cell therapy, and beyond. So if it could be helpful in kind of establishment of some behavioral items in your university, that would be very, very nice also for us, so we could transfer from our technologies. Speaking about this symposium, it was a little bit spontaneous, but I'm very pleased that so many lecturers were willing to contribute with their presentations. So today, for example, we have our presenters giving their lectures from also University, from Coimbra, Portugal. Well, myself, I work in Maastricht University, as well as in Moscow State University, Medical University, Tatyana University. So it's really very international event. And I have to say so that Kazakhstan offers truly international environment for collaboration and for promoting research. This symposium is supported by the small company Blast, associated with Maastricht University, as well as by two EU programs of staff exchange. So one of them is called FITA-UP, and it's focused on the studies of new ketone compounds and similar compounds which are designed to improve maybe conditions with neurodegenerative disorders, something Prof was speaking about. And the second one is called ASSAY. So this is a little bit more of environmental direction. We'd like to know how water can impact the health of a human being, and also in the context of neurodegeneration. So this will be a special session on the last day of our program. As those have mentioned, we have universities from France, Portugal, Netherlands, Russia, contributing to this event. And I hope that you will find it interesting, and please ask questions after our talks and during coffee breaks. And I wish you a very good time and interesting program and enjoy it. Thanks a lot. So it is my pleasure to present the first speaker of the symposium of today, Professor Daniel Anthony, who made all his way from his road to a little bit early way, but yes, to give a talk on the decided today on METABOLOME, studies with models and the patients suffering from neurodegenerative conditions. So Professor Daniel is our long-term collaborator in Maastricht and Moscow, but now he will continue working with us. Maybe also contribute to work in Nazarbayev. So the floor is yours.
Thanks, that's great. And this will just come back to life in a few moments. Okay, and I'll just share the screen on Zoom. Let's share the screen on.
Good start already.
I think that's going to be okay.
Yeah, it's good start already. Yeah.
Share screen desktop too. Let me get it share. Okay, well, I'm hoping everybody else online can see the presentation on their screen, and you clearly can because it's there. It's very nice to be here. It's a surprising place. I've been reunited with my bags as well, which I'm very happy about. There was Turkish Airlines is fantastic for getting me here, but everything was fantastic except that my bag only arrived this morning. But hey, it's here. It's brilliant. So my plan is to actually to tell you a little bit about a technique that we've been using to try and answer some difficult questions that clinicians face. I'm going to talk about a disease which is that you might not think of as being particularly neurodegenerative, but that was interesting. That's not moving forward. That's my problem. There we go. Okay, but you might not think of us being particularly neurodegenerative. I'll change your mind on that in a few moments, and I'll tell you about some of the issues as to why we care about this disease. Anyway, it's one of those that we started with a long time ago. I've just put up on the screen a set of the things that we do. So Tatyana said, hey, why don't you talk about metabolomics? And I'm not entirely sure that this was necessarily the best choice, but I thought that what I'd start with is just showing you what we do overall in my laboratory in Oxford. And fundamentally, I started off in the world of inflammation and inflammation in the gut, funnily enough, not inflammation in the brain. I decided that was really rather too smelly. And I decided to move into the studying inflammation in the central nervous system. And the reason we care or are interested in that is because it's so very different from inflammation in the periphery. So we all know what inflammation looks like in the skin, for example. If you end up with a nasty spot on your face or something, it's red, it can hurt, there's swelling associated with it. But in the brain, first of all, we can't see it. But secondly, those characteristics in the periphery, which are that change in blood flow, that extravasation of white blood cells, neutrophils, for example, that are in abundance in your blood, enter those sites of inflammation in the periphery, where they can cause some additional damage, where they can, but they're responsible for sort of clearing pathogens, promoting repair. And many of the inflammatory, the response that we're associated with, we associate with inflammation, for example, that redness is a consequence of the release of a whole load of molecules that are vasoactive, that cause tissue edema, that indeed give rise to further recruitment of white blood cells, again, to try and promote the resolution of whatever's caused that inflammatory response, whether it be infectious, whether it be trauma or whatever. In the central nervous system, it's completely different. You virtually never see a neutrophil in the brain unless you've got neuromyelitis optica, for example. Only very few neutrophils are ever recruited to the brain after a stroke or indeed after a head injury. There are no mast cells in the brain. There are no dendritic cells. There's no traditional lymphatics. And all of these things and all of these differences contribute to a very different place that makes it an interesting field to study, because from time to time, for some reason, the intrinsic nature, the protective nature in the brain is overcome, and you end up with a disease like multiple sclerosis or neuromyelitis optica. Or in certain circumstances, after an acute injury, you end up with driving an inflammatory response where we know it can contribute to the to poorer outcomes. So that's really where I started off. So many years ago, I went to Oxford. I guess I'm sad to say it's in 1994. I pitched up there, and I just got my PhD. And I worked with a guy called Hugh Perry. And at that time, there was one therapy for multiple sclerosis. Beta interferon had just been released. Now we have about 27 different therapies, and it's a very different disease, but it's still very problematic. Over the years, and I highlighted multiple sclerosis here. We do traumatic brain injury models. We do spinal cord injury models. We've started working a lot on cancer diagnosis. I'll show you a slide at the end of that. I've got a fabulous postdoc working with me who has an interest in the influence of the gut on brain function. He's interested in colitis. He's also interested in sort of in fatty liver disease. So we do quite a lot of that work. We work with guys in Brazil on arboviruses, and we also, by nature of this symposium, but I'm not really going to talk about it, we do a lot of we work on neurodegenerative disease models. That's what we like to do. Is we like to look at behavior and then relate the behavior to what's going on at a molecular level in the brain, and we can do that by using conventional PCR methods, by using proteomics, and today doing metabolomics. So that's what I'm going to talk to you about. Right, so you're looking at some brains here. When you look at these brains, you're thinking that's got to be a really nasty neurodegenerative disease, and that's got to be Alzheimer's disease or something like it. In fact, what you're looking at is progressive multiple sclerosis. So when people used to get multiple sclerosis, everybody said, you know, this is just going to be a little bit of motor dysfunction. And multiple sclerosis, which basically is an autoimmune disease that affects the central nervous system. And it's always assumed, for so many years, it was assumed that the inflammatory immune response was mounted against myelin, and it was a demyelinating disease. And if you could stop the demyelination, if you could encourage remyelination, the disease would go away. In the 30 years that I've been studying this disease, this has been turned on the head, and to some extent we were responsible for turning it on its head. We showed for the first time that actually there was a lot of axonal injury that happened as part of multiple sclerosis, and it didn't happen as a consequence of long-term chronic demyelination. It happened at the time that the myelin was damaged. You lost axons. In fact, downstream of that, we've shown that you lose neurons as well. So the immune assault on the brain gives rise to not only demyelination, but also the loss of axons and loss of neurons. And that's how you end up with a brain that looks like that. Now, as I mentioned, there are now plenty of therapies for multiple sclerosis, and some of them are really effective at preventing the relapse. So if you suffer from multiple sclerosis, what tends to happen is you have periods which are perfectly normal, and then you have periods of heightened disability, and they seem to get better again afterwards, but they never go back to baseline. And the reason they never go back to baseline is because you're losing neurons and you're losing axons. It is a degenerative disease. It is a neurodegenerative disease. Now everybody's very happy with that, and you can see why because you're looking at these pictures. But there are still plenty of problems left to us. First of all, what sort of disease does somebody have when they first present with demyelination? They notice something wrong with their motor movements. They have eye problems. This is how they present. But do we know whether they're going to be the sort of person that has a little bit of disability and then remains fairly static for their lives? Or are they going to end up with a brain that looks like this quite quickly? We know that if we give people therapy, those relapses will go away. But we also know that the time taken to when people make a transition from having a disease called relapse and remitting disease to secondary progressive disease, not all people do move on to secondary progressive, but about 50% of people go on to develop this sort of disease that then really rocks their brain. And when that happens, nobody really knows exactly when that has happened. And it's really difficult because if you don't know when that's happened, you don't know that you have to swap people onto different medications. And as I say, we don't know whether somebody's going to remain fairly static and live a life that isn't going to be much affected by the disease, or whether they're going to be killed by it, because lots of people die with MS. So we have adopted, and what I'm going to talk to you today about is a way in which we can interrogate biofluids in such a way that we can look at the metabolome and use some advanced mathematics, which enables us to understand whether or not there are signatures, unique signatures, to tell us exactly what's going on in somebody's brain. It's very easy with a rum punch. I can look at it. You didn't know it was a rum punch, but I'm telling you it's a rum punch. And the clever thing is you can taste it and drink it, and then you'll know that it's it might actually just be a fruit punch. It might have some rum in it. It might not have some rum in it. You certainly can't tell. But if you were worried about whether or not you could taste it and taste the difference, you could take that sample of this, which looks a little bit reddish. Let's imagine it's a serum sample. You put it into an NMR machine, and then we can look at the proton spectra of that. With that proton spectra, I'd be able to tell you exactly what sort of punch this was, whether it was a rum punch or not. I'd be able to tell you I'd probably be able to tell you what sort of rum was used. So in actual fact, bizarrely, NMR spectroscopy, so basically looking at all the low molecular weight compounds in a fluid, has such a unique signature that we can actually use it to authenticate something. You'll be astonished to learn that actually your own blood has a very unique signature. Where we've taken blood from lab members over an extended period of time, we've discovered actually their metabolome, their metabolic signature inside their blood, as measured using proton spectroscopy, is pretty much static. If you go out and you have a very, very fatty meal, you'll increase the levels of your VLDLs in your blood, but everything else, bizarrely, remains pretty much the same. So from that point of view, it's a really interesting fluid to be able to sort of analyze and decide whether or not we can see any other features that are in use, such as the presence of a pathology, and then whether we can say, you know, what it's doing, whether or not you're going to respond to a drug, et cetera, et cetera. And I'll give you some examples now of how we've dealt with that, and also highlighting how it is perhaps that we can actually manage to report on something that is going on in the brain by analyzing a peripheral biofluid, such as your urine or your serum. So many years ago in Oxford, we discovered something that was quite striking and surprising. I was telling you how the brain inflammatory response is so very different from a peripheral inflammatory response. And to that end, what's happened is that the brain is so different. So, for example, histamine that's bioactive in the periphery and will cause an increase in cerebral blood flow. In the CNS, histamine is a neurotransmitter, serotonin, that we always assume that's associated and was discovered in platelets. It's lots of it in the gut, for example. It controls vascular tone. In the brain, it's a neurotransmitter. Cytokines, I'm not going to say the neurotransmitters, but cytokines are responsible for controlling the inflammatory response. When they're present in the brain, they do different things and they modulate neuronal function. But in the periphery, they drive the inflammatory response. So we wondered, was it the case that the CNS inflammatory response was different because no signals ever left the brain? Normally, when you have a chest infection, for example, or a UTI, you switch on something called the acute phase response, which is an event that happens in your liver, and then this produces serum amyloid A. It produces C-reactive protein, generates IL-6, for example. We've shown TNF and chemokines. What we found was that if you injure the brain, you get dramatic changes in the liver and the spleen immediately after injury. In fact, you get those changes well before you see any change in the brain. So there are signals that leave the brain and change the way other organs behave. And I'm telling you this because I'm trying to explain how it is that a small focal lesion in the brain, and when we generate a small focal lesion, this switches on events in distant organs and it changes your whole body. And that is something that we got very interested in, and we've been able to show over the years how that mechanism works, how that signal gets out of the brain. And we've shown that it uses, it's very interesting, it isn't the release of a small low molecular weight molecule, even a cytokine. What happens is the brain starts releasing these things called exosomes. And the exosome leaves the central nervous system and switches on an acute phase response in the liver. And we've done some clever experiments, and with my friends in Norm Horkey and at Johns Hopkins, we were able to show precisely how that mechanism works, that neutral sphingomyelinase is very important in generating these extracellular vesicles, that there's and the actual nature of the response that's generated in the liver. So that's kind of a fun thing that we've done, but it's highlighting again, you damaged and you injure the brain and a signal leaves, and that signal changes the whole way that the rest of the body is working. It mobilizes white blood cells from the bone marrow into the blood, so they're then available to enter the brain. Here's an example of multiple sclerosis. And it's also quite striking that if we've got human control human liver, human multiple sclerosis liver, and then EAE, experimental allergic encephalomyelitis. This is the model we run for multiple sclerosis. We run some focal models, but we do tend to use this one from time to time. That's a control mouse that's just had a complete Freud's adjuvant injection into its legs. And you're looking at the number of neutrophils in the liver. Okay, so this is quite striking. This means this is when people think about multiple sclerosis, they always think it's a brain disease, it's brain and demyelination. Demyelination. And I've told you actually it's not just demyelination, it's axonal injury, it's neuronal cell death, but it was also a systemic disease. You're getting really rather large numbers of the brown dots on neutrophils being recruited to your liver, where they damage the liver and they switch on, they elevate things like serum transaminases. And it's a very surprising thing that this is, whenever neuroscientists talk, neuroscientists like to just think about the brain, but in actual fact, your whole body is changing as a consequence of having disease on board. I'm going to skip this description here, but it's really just to say that in the periphery, your peripheral immune system is responding to what's going on in the brain, and it's changing all sorts of things in the periphery. And we shall see precisely some other examples. So many years ago, we started off on this idea of, could we see a molecular signature for neurodegenerative disease by studying some small focal lesions? A guy called Jules Griffin, who is basically one of the founders of metabolomics and NMR-based metabolomics. And I went to a lecture that he was giving where he was talking about how he could tell the difference between two different types of water vole. Or a field vole and a water vole. Just to let you know, a vole is like a small rodent. It's a mouse that lives down by the sort of the riverbank. And he was able to take the urine from two subspecies of this animal, of these animals, and show that he could measure differences using their NMR spectra. I listened to this and thought, this is a completely stupid thing to do with a really expensive piece of kit. I mean, you can do it, but why don't we actually look and see if we can distinguish between different sorts of lesions in the central nervous system? If you inject TNF alpha, one of the principal pro-inflammatory cytokines, into the brain, it recruits a lot of mononuclear cells. When I say mononuclear, I mean principally monocytes. They turn into microglial cells. If you inject interleukin-1 beta, something rather odd happens. After a delay, initially, acutely, nothing. But after a delay, you do get some recruitment of neutrophils across an intact blood-brain barrier. The blood-brain barrier remains perfectly intact in both of these scenarios. But we get two different styles of pathology. One of the pathologies with TNF looks like multiple sclerosis. The other pathology with IL-1 looks like neuromyelitis optica, one of these sort of diseases that a disease that has been peeled off the spectrum of multiple sclerosis-like diseases. And what we were able to show is that in the urine of these animals, their metabolome was distinct. We could tell the difference. We could say exactly which cytokine we'd injected in the brain by looking at the metabolic profile in their urine. So that was the preamble. That was many years ago. Then we teamed up with a bunch of neurologists in Oxford and in Basel and in Finland. And we asked, now, we've seen that we can do this sort of thing in rodents. Can we do it in humans? And we decided that a big, strange delay each time with this, and I wonder whether anything's ever going to come up. But we've already talked about what we can measure. Now, the thing about urine is it's not very stable. Your urine metabolites change around quite a lot. So in actual fact, what we tend to do is we look at we use serum or plasma, and there's very little difference between whether you use serum or plasma. It really depends what the clinicians are prepared to provide you with. And as I said, everybody in this room, you have a unique serum, you have a unique signature. We would be able to tell the difference between all of you just by looking at your metabolic signature, and it remains fairly constant. With the human work we do, in order to try and distinguish between different types of disease or subtypes of disease, what we do is we use a system of mathematics. I say not particularly advanced mathematics, but it has to be done like this. We take a dichotomized group, for example. We generate some random permutations, for example, and we generate we split it in such a way that we end up with a training set and a test set. And we train on these models. We go through 10,000 iterations to make sure they're actually working. So what we do is we're looking at all the spectrum, and we're trying to build models that are able to separate using, to start with principal component analysis, and then OQL-SDA, sometimes random forest. Our mathematics does change a little bit, but the whole point is we have an independent test set. In everything I'm going to show you, there's always an independent test set that hasn't been used to model build. And then we work out an accuracy by putting that into the algorithm that we build. Right. So, and I'm sort of glossing over this a little bit because I am actually a biologist, biochemist, not a mathemaician. And I work with some very clever mathematicians who would spend much more time talking about math than not talking about the biology. So here is the sort of a set of problems that we know exist in the field of multiple sclerosis. These are all questions that we'd like to ask. If somebody has a first sort of demyelinating episode, and we don't know whether they're going to move from something called clinically isolated syndrome to full-blown MS, that's a question. Is somebody going to do that? I said right at the beginning that we need to know those sort of things. Then, if somebody has MS, are they going to convert from relapsing remitting MS to secondary progressive MS? That was a really big problem that all our clinicians said they wanted to do. We also want to know whether or not people are suffering from subtypes of MS, for example, whether they have neuromyopathy or MOG-mediated disease. Within the sort of the neuromyopathy spectrum disorder, there's MOG, aquaporin 4 mediated, and sometimes people who have seemingly this strange NMO type, but we can't find any antibodies. We don't know what they are yet, but we've managed to generate metabolic signatures to show that people have this sort of disease. You'd like to be able to select the right therapy and know whether somebody's going to respond to it. Say predicting stability is very important. Predicting progression is very important. And I'm going to show you some of the things that we've done to illustrate that. So, firstly, can you tell the difference between somebody who has relapse remitting or secondary progressive? And this is where we basically started out. And we've now done this 4 times with 4 independent cohorts now. So still, there'll be a training set and a test set. And we've got to the point where if we look at their blood and we're looking at these plots, these will appear throughout the presentation. This is principal component analysis. And what we're doing here is we're showing a constellation of metabolites, not just one, but a whole bunch of metabolites. When we generate the algorithm altogether, we can find a pattern. And that pattern is able to tell the difference between those people with relapse remitting MS over here and those people with secondary progressive MS. And we can now do that with an accuracy of 91%. That's pretty good. That's enough to make some really good clinical decisions on. And other things that we've been able to show is that we can beat people, we can beat clinicians in terms of deciding whether somebody has got relapse remitting or secondary progressive. So this is an example of an individual where we were able to show that they had transitioned. We were able to show that transitioning before a clinician had spotted it. So the biochemistry, I'm afraid, can be better than, I'm now preaching to converted here, but biochemistry is often better than the clinicians. They don't like to admit it though. Here's another example, this is effectively addressing that issue as to whether or not somebody really has got multiple sclerosis or not. And so right at the outset, we can take our generate an area under the curve here of 0.88 in order to be able to show that we can actually diagnose those individuals with multiple sclerosis. Now, one of the things that we like to be able to do is mix and match. And one of the things that you can do here is you can take a proteome and you can mix that with a metabolome or a lipidome. In this case, we're taking a metabolome and mixing it with a proteome. It's simply a question of just expanding your database, more columns on your database, and you just have to work out how to normalize your data. And it's not that complicated, really. But what we're able to show here is this is actually the diagnosis of multiple sclerosis. Now, you would say, aren't clinicians good at doing that? They're not so good at perhaps at establishing whether how quickly somebody will go from CIS to MS. But one of the things that is happening in the world of multiple sclerosis is that people are massively overdiagnosing MS now. Because if you have the presence of something called oligoclonal bands in your cerebral spinal fluid, this is often used to diagnose. And it's an unsafe way of doing it, because in actual fact, it massively overdiagnoses. And what we were able to find is when we analyze using something called SomaScan, it gives you 5,000 proteins to look at in the CSF. And we would then mix that with our metabolome, and we generated a multivariate model that was able to very accurately predict precisely whether or not somebody has multiple sclerosis. And coming down with these very, very high accuracies, almost 90%, using CCN5, von Willebrand factor from our metabolome myoinositol and the oligoclonal band status, we're able to say whether somebody has MS or not. So it takes a long time to get these things implemented in the clinic, but we push and talk to our clinician friends about why this is going to be important. And then one of the things, obviously, that we've had a go at, and it's a useful thing to be able to do, is predicting whether somebody is going to respond to therapy. And again, we've managed to generate some algorithms. This glutaramate acetate is a first-line therapy for multiple sclerosis. Some people will just not respond to it, and they will then wait a year to find out whether they should be switched to another therapy. And in England, after they've decided to switch, it often takes another year for them to actually switch, because they have to have some MRI scans, they have to be reported. It's a crazy long time. So in actual fact, if you could just say right at the outset, I give you my blood, I'll tell you whether or not this therapy is going to work or not right at the outset, and this would be a good thing. So We played this game with multiple sclerosis. We've done it for some other conditions as well. So I'm conscious of time, and I'm going to scamper through some unpublished results now. So one of the holy grails that we think we've absolutely nailed in multiple sclerosis research is being able to tell somebody whether they're going to progress, whether their disease is going to get worse. And so you want to know whether or not somebody will actually end up over the course of the year, whether or not, independent of whether they're on a drug or anything else, we'd like to know, do they have disease that will progress, independent of whether they're relapsing, remitting, or secondary progressive. And so we've taken a large cohort of individuals from our colleagues in bloods from our colleagues in Finland, Laura Aras, and she has scanned all of these individuals using a PET ligand called PK11195. This ligand binds to activated microglial cells and astrocytes, and she has shown that the binding of this PET ligand predicts whether somebody is going to progress. But PET scanning, when I do it in Oxford, it costs 2,000 quid a mouse, basically. This is not going to become common for monitoring people with multiple sclerosis. But knowing that actually there's a signature here that can predict progression, gave us the idea of saying, well, in that case, can we actually manage to see a metabolic signature for that progression in their blood? And so what we have done is we looked to see initially whether or not we could spot the difference between the high and low inflammation that exists according to TSPO binding. And we stratified individuals either being low or high. And then we decided, and then we looked to see whether or not we could actually generate a model that would see whether or not we could predict that high and low TSPO binding. And we found that just three metabolites, glutamine, glucose, and glutamate, the levels of these three metabolites was able to predict whether or not they would be in the high or low TSPO binding. Now, moving on from that, a lot of cross-validation was required, and then we looked to see whether or not if we took our three metabolites and then worked backwards and said, okay, can we now look and see whether or not we can predict who's going to have progressed on the basis of the clinical scoring scheme? So now not the same as the TSPO binding, but actually have they physically progressed according to whether a year later when a clinician saw them again. And once again, we can do that with an accuracy of 0.78. Now, what's interesting about that is that that's actually the same accuracy that TSPO binding achieves. So we are as good as TSPO binding. You might argue that's a bit circular because, of course, we were building our models off the PK995 binding. What was then interesting, Daniel Radford-Smith said, what would happen if we mix PK11195 binding with these metabolites? And we got the benefits of both the negative and predictive value that come out of our models. And now we can say with an accuracy of 0.98 whether or not somebody is going to progress in the year. Nobody has ever managed to do that and predict progression in that way in multiple sclerosis. Now, sadly, this does again use PK11195 as having to give somebody a PET scan. But there may be, as we hope, that there'll be other indices that we'll be able to use. And we've teamed up with Novartis, and they're making 8,000 MRI scans available to us in order to see if we can find other markers from an MRI that we could then use and mix and match with the metabolites. So this is quite game-changing. The other interesting thing, just to note, is that largely these concentrations of these metabolites that were interested in glutamate, glutamine, and glucose were pretty much independent of whether people were getting these different therapies. Some of the therapies, teriflunomide, for example, did seem to move some of the biomarkers around, but it also has to be remembered that none of the therapies really that are available, none of these ones at least that are listed here, are actually any good at treating progressive disease. So perhaps it's no wonder that these drugs don't actually affect particularly the metabolites that we showed that are good for this. Right, so what's interesting now is if we get back to think about one of the biomarkers that's used to look at neurodegenerative diseases are neurofilaments and neurofilament light chain. we looked at, this is neurofilament heavy chain, and we also measure neurofilament light chain in blood. It's really useless, actually, at predicting what's happening in the brain of an animal with MS-like disease. Actually, it's really poor. It does go up, but it also it goes up after a delay. It's a pretty useless biomarker. What was interesting about the result that I just showed you is that when we look to see how axonal injury, so basically progression, how it correlates with these, this is a completely independent study. And we found that actually, once again, it's glutamate and glucose, where we've got really quite large R values, showing that those metabolites and their concentration in blood is very much correlated with the amount of axonal injury that we get. Good, okay, so that, I'm going to stop talking about multiple sclerosis now, and I'm just going to quickly show you some other examples which are to show how this can be applied to other conditions. This is some work we've done taking individuals where you're not sure if they have cancer or not. They keep coming to their family doctor and they don't know, and the family doctor doesn't, so they say, well, that's not breast cancer, it's not colon cancer, or I don't think it is. I don't know what to do with you. I'll do lots of tests and I'll send you around and around. We've taken the blood from those individuals. And these are individuals, there was a special study that went on in Oxford a little while ago called the SCAN pathway, where if a doctor was concerned about an individual and thought they might have cancer but didn't know what sort of cancer, they got sent for a whole body CT. It was then reported by a number of different radiologists. And we took those bloods and we discovered that we could actually, again, with very high accuracies, we could determine whether or not individuals had cancer, not just a cancer, any cancer. Now, you could say that that's problematic because then where do you look? But the point is that if somebody is going to have a scan, and most scans are negative, when you worry about somebody having cancer, you send them up to the radiologist and they say, well, I can't see anything here. And huge numbers, I think about 90% of people who are scanned have absolutely nothing wrong with them whatsoever. Now we're able to say, this patient does have something wrong with them. You've got to find it. And when we've talked to our radiologists now in Oxford, they now look at those individuals who have this blood test and they find the cancers. We're hoping that downstream, we will be able to say whether they have cancer and what cancer they have. So that's something that we sort of, again, turned our attention to on the basis of those initial results of multiple sclerosis. This is something completely different. This is kind of interesting. People who are psychotic. Some people who are psychotic have antibodies against receptors in their central nervous system and different proteins that are on the surface of neurons. And what we've been able to show is that for certain antibodies that are present in the circulation, we can see a very distinct profile of individuals who have antibodies against BJKC or their glycine receptor. Kind of strange. This is clearly a different sort of psychosis. We can separate them, and it's a different sort of psychosis. And in terms of them, when we drill down on this particular population, we discover that their symptoms are also slightly different. But it may be that these sorts of people who basically have an immune-mediated disease, potentially, that actually you could actually use plasmapheresis to treat them rather than having to use antipsychotic medicines. Right, another last example of things that you can do with NMR in the context of neurodegenerative disease. So going all the way back to the originally, there's that original description of what we did with the metabolome. This is TNF into the brain. And what we're looking at now is we're looking to see, this is some PET scanning showing the uptake of FDG. And we can say that if we put TNF into the brain, we get an increase in the uptake, an increase in metabolism in the brain. But where is that metabolism? Is it in neurons? Is it in astrocytes? What does this actually mean? Now, using NMR, what we can do is we can show immediately, as we already know, that we can actually see the difference between a saline and a TNF injection. That's something that I started out by showing you we could do. But now let's say that we label the glucose this time with carbon-13 in 2 different places. And this time we can get a bit more funky with our analysis, because what this enables us to do is it enables us to actually track metabolism and understand which cells are metabolizing more or less and what's happening in the brain, how that glucose is being utilized. So these are sort of the suspects who were thrown up, showing that in the TNF that our carbon-13 glucose double labeling increases alanine and glutamine levels, for example, in the brain, and decreases creatinine and myoinositol. And from there, we can show that in actual fact, that what we have been able to show is that we're selectively activating the pentose phosphate pathway in astrocytes. Now, why does that matter? Well, the problem with activating selectively the pentose phosphate pathway, which basically is an anabolic state for the astrocyte in order to start become activated, and then sort of engage in proliferation, engage in doing things other than supporting the neuron. And what we were able to see is that neuronal metabolism was either unaffected or slightly decreased. Glutamate levels weren't changed, but what we ended up with was a scenario where it looks like the astrocyte is basically going off and doing its own thing. And those of you neuroscientists among you will know that basically an astrocyte is there to do involved in neuromodulation, but also to be an energy source for neurons. And what we've shown here after the TNF injection using metabolomics is that in actual fact, the astrocyte stops supplying the neuron with energy metabolites and leaves it on its own. So in terms of thinking about interventions, what we need to do is we need to start thinking about whether or not we can manipulate the behavior of individual cell types, and in particular, the astrocyte in the brain. Good. So look, I'm going to call it quits there. And thank you for listening. And I hope that to some extent, what I try to show is how we can take neuroscience and we can apply some biochemistry to it and some mathematics to it. In actual fact, when you're harvesting your tissues, there's an awful lot of information in there that you might be able to sort of get some really interesting and useful insights into what you're doing and how your system is changing using tools that you hadn't perhaps thought of using. So there's a huge number of people that have done this work in Oxford and abroad and elsewhere. So I'll stop there and take any questions. Thanks.
Professor, thank you very much for the interesting lecture. So I would like to ask audience if someone has questions.
Thank you, it was very interesting talk. Actually, I was quite surprised that metabolic signature is quite stable for the same person, because I was thinking if you have some kind of obesity or diabetes or liver diseases, or even when you take some drugs or alcohol, these parameters will be changed, and you found that it's extremely stable. How it happened?
So what the problem is we have a lot of not a particularly large lab, but a lab that's large enough and then when I say they're lab victims, I think is perhaps a better name for the lab researchers in our lab. So we serially bled them, but we also had interventions. So they were forced to, on one particular morning, we took blood, then they all ate a McDonald's big breakfast, a huge big breakfast. And then we took the blood half an hour later, and this was totally astonishing. But we were trying to decide whether we had to impose restrictions on blood. So in the clinic, when we were collecting for the cancer study and for the multiple sclerosis studies, we say, well, are we going to have to insist that people fast, or we'll have to take it? And from these sort of studies that we were doing, and then following up a week and a month later, the same individuals, and discovering actually that there are basically no changes. It is quite striking, and we did not predict this. We thought that it would make a difference, and yet it didn't. The other thing that doesn't make so much of a difference is most of certainly the high molecular weight therapies that are used in multiple sclerosis, so antibody therapies, for example, they don't show up. And they wouldn't show up in these spectra because you have to be micromolar, millimolar concentrations in order to see anything. So anything that's there in a very low concentration, first of all, it's not going to appear in the spectra, but oddly, it doesn't seem to change it in the way that you'd expect. So these we call them type 2 biomarkers, because they are really reflecting the disease rather than what the intervention is doing to the disease that may or may not have an impact on the pathogenesis. But yeah, it does seem to be surprisingly static. And we did quite a lot to find out whether that was the case.
Just to follow up, thank you very much for a really nice, exciting presentation. So, Daniel, I have a question about maybe just follow up with Gabe's question. So, maybe because of sample preparation or just because using NMR, could it make a difference? That's what you steady state.
Yeah. So, I think what we've always tried to do is best case scenario. So we take blood and we spin it at exactly 30 minutes. But then we have done quite a lot of experiments where we have, which we've published, where we've basically done it badly. So we've waited 24 hours, we've done freeze thaws. Every time you, everything you'd expect that would probably not be a good idea, for example, leaving blood on the bench for 24 hours or going through a freeze thaw does have an impact. But oddly, these are the biomarkers. So when we do it with a perfect, so it's, for example, the glucose, the glutamine and the glutamate, those are the biomarkers. And all you're ever going to do is you're going to erode the signal, but you're not going to change the biomarkers. So the biomarkers are independent of anything you ever do. But your ability to actually discriminate or see those biomarkers changing might be affected. So we always try to be as careful as we can. Oddly, for the cancer study, the blood was collected very badly in the study, and yet it still gave us models where we could see cancer and no cancer. So it will also be the impact of the pathological process. If you're looking for subtle things, you'll probably have to be more stringent about how you collect and handle the tissue. And then if something is going to have a profound impact on your metabolism, and for the majority, for a lot of people, of course, when a physician is worried about whether they have cancer, it's whether or not they're cachexic, for example, and so that sort of profound weight loss you get with even tiny little tumors. So yes, I would recommend. You have to be careful, but the whole idea is to roll it out and make it a real clinical test at some point.
But then also the follow-up. So why did you choose NMR instead of mass spec? I mean, mass specs are more sensitive.
Absolutely. And we do mass spec as well. And there is, and on one of the slides, there was when we were looking at the treatment effect of deuterium acetate, we compared mass spec with NMR and pulled out similar metabolites. The difficulty with mass spec is, I'm afraid, we found that it just, in terms of, it's incredibly sensitive, but the throughput is very slow for this sort of discovery biology. If you know what you're looking for, and say I chose 3 metabolites or something, and I could do mass spec, and that would work quite well. But for assessing, for doing, first of all, there's quite a lot of the metabolite profile, whether you're using GCMS or LCMS. You're processing the sample in a slightly different way. The beauty of this is you simply take the blood, you put it in the NMR machine, and that's it, it's done. So and it also gives you sort of quite an interesting wider spectrum of the molecules you can look at. For example, you get the lipoproteins, and you get but as you say, the problem is that the concentration is a bit higher.
So you go for NMR.
So we think that NMR is the one where, because it for a clinical service. It costs about £20, €20 to do this clinically. It's nothing.
So, and one more thing, just now it's more like science. So, you mentioned that in MS, multiple sclerosis, demyelination is probably a major problem, correct? So I'm not saying that there's definitely demyelination, but the difficulty is that this obsession with demyelination has caused people to focus on let's remyelinate the brain rather than let's stop the neuron from dying. And so, but my question is, like, if we're talking about this one of the criteria, right?
Yes.
So it's probably supposed to happen in the spine before it gets happens in the brain, and that's what
I think that's a fair. So in terms of, it's fairly random where the lesions present. So you end up with a clinical radiological paradox. So you can see somebody who has lesions all over their brain by MRI, but actually no disability. You only need to have one lesion in the wrong place in the spinal cord, and this will give you. So yes, it's very dependent on what happens in the spinal cord, and all the original early pictures and descriptions of MS. Very much concentrated on spinal cord, but spinal cord doesn't lend itself to PET or to MRI particularly. So that's why we spend so much time worrying about what's going on in the brain. But demyelination, absolutely, it's a feature, but it's not necessarily the target immunologically that everybody thought it was. I think that's the take-home message.
Okay.
Thank you.
Thank you. The discussion basically can continue on our social link and. Exactly. So let me present to you our next speaker. Basically, she will start introducing herself, but I will do it. So our next speaker is Professor Tatyana Strekalova. She is professor of the section of medical university of Moscow, and she's also senior scientist research at Prof University and Neurodegenerative company for the Netherlands. Showing the amount levels of the building, and today is going to be some levels of how do you
Right. Thank you very much. Sholpan. Thank you very much. I'm more maybe behavioral scientist per se, so maybe I will not be too explicit about animal models of neurodegeneration, where we happened to us that we are working a lot with this model last years, but fundamentally I'm more behavioral scientist and also to neurobiology. So this is my field. So my lecture will be a little bit unusual, as we have a conference / workshop, so it will be more about practical things regarding behavior. Because before here in National Word of Astana, have a book, thank you very much, plans on building the all animal facilities and establishing methods of behavioral tests and memory tests in particular. So I was asked to give kind of overview on the main techniques that are used to study cognition and memory in mice. So I will speak about mouse models of memory and how it can be used in the graphic of development. So mostly this work has been done in Germany and Netherlands. Should I use microphone? Yeah. And currently Maastricht University is my main location, but also the global help in the background like from the one from the base. The battery is this one. The technical things they don't like. Otherwise, they can use. All right, just to start with, so learning and memory, very unique magic properties of our brain, and basically we are what we have learned, what we remember, and what we forget. Where learning is a process of accumulation of knowledge, acquisition of knowledge, and memory is a process, thank you very much, of retaining this acquired traces, reconsolidation, and forgetting. I can see it on the screen, something happening, but so just next. Now. Switch on this one. Yep, it's gone. It's not changing.
It's not changing. Okay. That's something that we are not.
Fine. Can you
Switch them here?
Yes.
So if you turn the button, which one? So just go on. Not a problem.
Just restart the slideshow. Then go.
Okay. Connect. Here. This, yeah, no, it's
I know. Sorry. All right, so today I'm going to talk about what memory is, why it's important to study neurons, and what types of memory how we can classify it, can be studied. What are the main urban guidelines to study memory in cognition lab? What are the recent advances in studying memory? And as a separate topic, I decided to talk a little bit general basics of behavioral work in small rodents, because I think it has its specifics, and I do think it also could be useful to you if you are planning to start your own behavioral studies here. Right. So memory can be classified, and you will probably find many classifications of memory as to declarative or explicit or non-declarative or implicit memory. Declarative memory is a memory for facts, events, and as evolutionary younger form of memory, it can be in the first place during various forms of neurodegeneration and other medical conditions. Non-declarative memory includes skills in writing, as well as classical conditioning, Pavlovian conditioning, non-associative memory. If you look at neurobiology behind these points of memory, you can see that it covers different various areas of the brain. With declarative memory, it is known that the hippocampus, so in humans, and temporal lobe are critical. For instance, in mouse, it would be prefrontal cortex and hippocampus, to critical brain areas. With now it's good. So with associative learning, Pavlovian learning, as for instance, amygdala, hippocampus, cerebellum are very important. But today we are not going to address this molecular cellular mechanisms and circuits underlying different types of memory. As I said, it will be a little bit more practical and concerning actual existing memory tests that people probably use in the near future. Regarding other types of classifications of types of memory, it's important to say that memory also can be regarded as working memory. So this is memory of living creatures, us during performance, execution of particular tasks. Then we have short-term memory and long-term memory. So long-term memory, and memory process is lasting 1 to 3 hours, and whatever takes longer is memory processes are 24 hours, definitely considered as long-term memory. So why do study learning and memory nowadays? First of all, of course, they have exactly the basic interest in how learning and memory are organized. Then we like to understand the mechanisms of cognitive decline when we have patients clinic suffering from different forms of cognitive problems. And this will be in the first place various forms of dementia, and of course, disorder. And with groups here already actively engaged in this type of research. For example, Prof. Sholpan studied microbiome in patients with psychotic disorder, and Prof. Tatyana has quite some work with the disease, studying environmental factors, potential bacteria
criteria for other protective interventions. Well, yes, memory is impaired during aging, during various neurological, psychiatric conditions such as depression, for example, and then they also be impaired during somatic disorders. For example, when we have compromised hormonal processes, metabolic processes, medications, and such. So yes, so we like to study memory in the lab because we like to offer better maybe pharmacotherapy and other forms of CME memory impairment. Then, as I mentioned, more and more important now, I think in research to understand how the environment can actually influence the functions of the brain of memory cognition and how we can actually prevent this potential negative effects. So, as I mentioned, there is this whole list of medical disorders which contribute in the first place to a cohort of patients suffering from cognitive problems, and exactly this is what standing in the first place. Later on, we will hear more about what we do in this respect. To give us very more give us an overview of what our laboratory is doing in this direction. Right. So clearly, we have a deficiency in availability of proper means, remedies in treating cognitive problems in patients, and this difficulties can stem from also insufficient quality of our work in the lab. So when we talk about behavioral assessment of potential drug candidates, memory enhancers that can be used to improve cognition, resolve memory problems and patients. So I'd like to emphasize that actually we work here to deal with just one element of learning and memory, where the whole learning memory process is constituted from acquisition, first of all, of memories, and consolidation, storage of memory traces, and then recall. So basically, when we talk about memory tasks, lies all that. So we're dealing with the recall stage. And we also have extinction of memories that we will discuss this issue today. What are the main laboratory methods to study learning memory in mice? So if we refer them to different forms of memory, so we have these lists which are probably tests which are probably used on the world. For example, working memory is frequently studied also for fly maze, the partial form of the test. Barnes maze, noise with maze. Short-term memory is frequently associated with step-down avoidance, peer conditioning, object recognition tasks. For long-term explicit type of memory, people use a lot also these tests that I just listed, so down avoidance, peer conditioning, fly maze, voice recognition, Barnes maze, as well as object conditional-based diversion, social transmission of patterns, place preference. In addition, many researchers like to study instrumental learning more than right. And for that purposes, the frequently used paradigms are shuttlebuck, random pressing operand tasks, and similar model is based on this instrumental operational law. So for preconditioning, animal is trained to associate aversive context with electric shock. That is painful, but normally it's a slight shock. So the context in which it receives a shock is perceived as aversive to mouse, and it responds to this unpleasant stimulation with freezing behavior, because in nature, mice naturally they freeze or they want to hide from a predator. So what we measure in this test is the duration of freezing response. So this form of learning considered to be hippocampus dependent, and it is frequently used to characterize various little mice in which we expect to have problems with hippocampus dependent long-term memory. Auditory recognition. In this variant of the test, mouse is exposed both to the food shock and auditory stimuli. Whether this form of learning is quite distinct, it's not even auditory dependent. So again, mouse, when it is exposed to, repeatedly exposed to the sound, when it records the sound, it will display freezing behavior. Step down avoidance. So this is another form of contextual learning. And mouse is supposed to learn the association between safe platform within the context of this box, in which it also receives a slight shock. So when it is placed with this box, again, after this pairing, when it remembers the context, it tends to remain sitting on the platform. So this is also a very common test for the hippocampus dependent memory, which is also used in various labs. Morris maze. This is another very common test, which, depending on the modification, can be used to study spatial memory or instrumental memory. With the spatial form of this test, mice are placed in water and they are allowed to find a platform that is kind of a safety area of them, but they're supposed to do a spectral map of the area around them. And for that, so they would use the visual cues. So this form of memory is considered to be hippocampus dependent, and it's also this test frequently used in laboratory science to study hippocampus dependent memory. At the same time, the instrumental version of this test allows to study mode memory. So you don't put visual cues, but you allow a mouse to know that there is a platform hidden on the water, which it can find once it just make a circle. So it will just randomly get on the platform. Okay. Bias maze. So this test is considered to be like a dry version of Morris maze, meaning that mice are exposed to visual cues, and the area which is a little bit unpleasant to them, the area, but they can escape it by the hole to hidden to home page. And again, the idea is that they will kind of special memory and navigate on this surface of bias maze to find the right home. Wire maze. Similar to voice maze, it can be spatial form of this maze or instrumental variant of the maze. With spatial form, you put again the visual cues, and mice is supposed to identify these cues, find the right, to choose the right arm where they get kind of rewards. So that can be water if they are deprived. It can be a bed, such as and food if they are food deprived. With the instrumental version of this test, mice are trained to change the sequence of movements to get a reward. But then no visual cues be involved in protocol. No visual. The social transmission of food preference is a sophisticated test. Mice can learn a safe sound from their common partner when they are food deprived and interact with a mouse that was fed with a scented diet. And then this mice is given a choice between diet with familiar flavor or unfamiliar flavor. If they learn the task, they choose familiar flavor. But this is quite a sophisticated task to run. Object recognition, another test, which is broadly used in laboratories. So, mouse is exposed to two identical objects in the first day. But by the way, it was a real object that we found work very well. So it has to be kind of interesting for mouse to have a complex surface. And then on the second day, you replace one of the objects with a new one. And in case the mouse would remember the previous object, it would spend more time exploring novel objects. So you can calculate the index of discrimination that would be a parameter of memory in this case. We also use a lot in the lab now, so-called condition-based aversion, where mouse is exposed consequently to a paste of sucrose, given it sucrose solution, and quite unpleasant injection of weakening peroxide that forms erosion. And in case if an animal builds this association between these two situations, next day it will avoid the sucrose. So, well, there are plenty of variants of instrumental learning, which is very much dependent for this type of memories on sophisticated apparatuses. And they include level pressing tasks, shuttle box, and basically this part of operant behavior is based on the ability of mouse to associate certain signals, such as light, for example, with some possibility to get a reward, or possibility to avoid multi-generative. These methods are quite demanding in terms of finances and also time. Now, I promised to talk a little bit about advances in behavioral methods, memory learning models. I don't know whether you would consider this as something advanced, but in our lab some time ago we built a machine that would allow to train animals in an automated regime to perform certain tasks. So we thought it's quite important to make to provide such conditions when animal is not in contact with us experimenters, because of course it creates stress. So we built a machine that was comprised of 20 pages, home pages, and they were equipped with sensors. So in the sleep there. And basically you can program the feeding regime or water supply in such a way so the feeders and this water supply spots could become accessible only when the mouse would perform certain movements in each. For example, moving from one position, position 1, position 2, certain area in the page, and making certain movement, for example, to read, making a reading. And only when it does all the sequence of this movement correctly, it gets rewarded. So basically, you can teach dancing from mouse, and it really works. And it does work in absence of physical contact of experimental and this is very important. And finally, what else is another thing that is becoming more and more popular, especially in the Netherlands, like the ability, for example, in neurodegenerative. Those are different models of virtual reality. So when the mouse is getting fixed on the wall, it can move, but also it exposed to images, and it can be wired to computer, so one can provide some simulation on the brain or to do some recordings. So this is quite a sophisticated system, but it's really, really helpful for neurological studies. Good. So this is basically the main part about memory tests, but I promise to say a couple of words about specifics working in the behavioral methods and principles, since you are going to start your own behavioral lab basically very soon. So with behavioral methods, we think behavioral work, you don't need the positive control or negative control kind of reference group. And this has to be included in every experiment that you are going to take on, rely on historical data to develop. You have to be sure that what you're observing as a memory or functionality, it is really essential. Then you have to be prepared to get paradoxical results, because sometimes you can't control all the conditions you're using. And then, if it happens, maybe the most trivial explanations have to be considered first. But still, so it's really wise to think about possible compounds with the tests you are going to use and try to get them under control. And of course, then to maximize the randomization of your groups with the study design. For example, just to give you a couple of examples. So frequently, you can observe increased locomotion in the open field or also maze. However, this hyperactivity, hyperlocomotion, can be a sign of low stress and low anxiety. At the same time, hyperactivity can also be a sign of panic behavior, impulsivity, and stress. So meaning that you have to be really careful about interpretation of the results and making observational parameters. Drinking behavior, so it's a very basic thing. You can find variability in the individual variability, not only in the behavior, as a behavior, but it's really remarkable that even very basic physiological functions can be extremely valuable within the group. We performed a very simple experiment, just looking at how mice drink during the day. And we found that, like humans, some of them more active in the morning. Some of them are more active in the evening, or majority will be more active in drinking in the morning. But it just, if you think about the tests which are based on drinking relation, drinking behavior, evolution, for example, if you randomly get in small groups majority of animals drinking in the morning or in the evening, you can actually run into false results. So then you have to bear in mind that we all have anthropocentric approach in the interpretation of mouse behavior. And for example, restricted mobility activity to mice means stress, it is stressful. Whereas for us, it's mainly relaxation, it's natural, the state of natural relaxation. So it's something we're searching for. Tail suspension, not something we were talking about today, but in the nature, mice like to be by the tail balancing in wild nature, but in laboratory life, so tail suspension is considered something very stressful for mouse. Yes, we should not forget that not all manifestations in behavioral repertoire or mouse we can interpret. For example, mice tend to choose from which side of the cage they like to consume the water. And it's very difficult to find out the reason for that. So there's no explanation. But yeah, the side preference has a huge impact on the evaluation of drinking. So then we have another test, yes, another test to call value displacement test, marble test. And I think even Robert Dickens from Oxford offered this test some years ago. He also has difficulty explaining why mice like to take out small objects from the tube. But it's important that they only do that when their hippocampus is intact. And they stop doing it quickly when they have impaired hippocampus function. But again, we cannot explain these behaviors. We don't understand why mice are doing this. Then we have some metaphysical factors in our behavioral work. For example, if you run the sucrose test, it can sucrose preference test. So where mice choose between sweetened water and normal water, and they always prefer sweetened water. All of a sudden, it's the very same bottles, the very same sugar, they might stop doing, and we don't have any explanation. So what could be the reason for that? Maybe the bottles are washed differently and the detergent was changed. So mice can smell it and can start to avoid bottles. Right? Maybe the sugar absorbs some cells. So all those things are absolutely metaphysical. We can't really think about it in the first place, but they happen. All the solutions of in the bottles can be cooled. So then you lose more just the physical factors when you put the bottle in the cage. Well, so we have to be prepared that some unexplainable thing can happen with some behavioral tests. Or when we use very bright light to denote margin light. So we increase the brightness more, and they like to hide the point of the open field, for example. But at a certain time point when it increases even further, they paradoxically tend to stay in the center. Why? Well, it turned out, so we also had this problem in the lab, they don't like reflection from the wall. So you always have to be really conscious about all these physical, metaphysical, physiological factors which have nothing to do with complex who will process it within the study, which is memory. But there are solutions to that. Well, we have to try to validate our parameters really well. We have to try to have a reference group of the setting in each experiment and try to identify possible compounds and possibly strict control over those with compounds. And what is very important, to use not just one, but multiple tests to study certain behavior, such memory, for example. So we can't rely just on one test. We have to use 2, 3, 4 memory tests. This is all. I'd like to thank you for your attention. And our groups will be started in Mannheim in Germany. This is Maastricht, and this is Moscow.
Since we are running about time, I think we can discuss during our. So we can now, since our Professor Alexander Tatyana is waiting online, and it's possible to we should continue to do that first.
Alexander Tatyana, who is Dr. Alexander Tatyana, but he will be studying in the thesis soon in Maastricht University. He's very experienced in behavioral work, mice and rats, and has very rich background dealing with as well for learning and memory a lot. But today we will speak about the model of neurodegenerative disorder that is amyotrophic lateral sclerosis. So he will speak about the role of inflammation in this disease and in this model.
Александр, вы нас слышите? Можете подключаться.
Hello. Good afternoon.
Но мы вас не слышим.
So you don't hear me?
А сейчас
Let's try it.
слышно?
No? What about now? Do you hear me?
Просто не заключила причина.
Do you hear me now?
О, да, сейчас мы вас слышим. Можете запускать демонстрацию экрана.
So.
It's our end of this. He just
Do you see me? Do you see the presentation?
We can hear him too. Можете еще раз сказать что-нибудь?
Do you hear me?
Not again?
No?
А почему мы не слышим? Мы же только что слышали.
was it from your speech was
We checked it today. Can you switch off the sound? Or you...
excuse me, you want to some good speaker?
No, it's switched off here.
Эти колонки не работают. Как сделать? Мы подключились в Zoom, и мы не можем. Тут какие нужно выбрать динамики? Наушник 2, вот он стоит, и мы не можем слушать. Может, колонки где-то отключились? А микрофон работает? А если отключим? А сейчас?
Okay, I'm trying to speak. So you don't hear me? No?
We can hear you now.
Excuse me?
We're getting some feedback.
Просто один момент.
Мгм.
Но нас-то слышно, да?
Мгм.
Mute that, we just asked you to start.
Не слышно.
Still don't hear me? No?
Go ahead when you want.
Yeah, so now you hear me. Okay. So let me share my presentation with you. And here it goes. Okay, so first of all, let me thank you, the organizers of this conference and the Nazarbayev University for hosting this event. And it is my pleasure to present you my latest findings, my work, which focused on the association of lipopolysaccharide, exacerbated neuroinflammation and behavioral disturbances in mice with truncated fused in sarcoma protein and FUS. And as you may know, FUS is a crucial RNA DNA binding protein that plays an important role in maintaining cellular homeostasis. And the truncation of this protein is implicated in several neurodegenerative disorders, such as amyotrophic lateral sclerosis and frontotemporal lobar degeneration, FTLD. So first of all, let me say a couple of words about these disorders. And FTLD is characterized by selective degeneration of frontal and temporal lobes, which leads to progressive dementia, while amyotrophic lateral sclerosis is a neurodegenerative disease that affects the motor neurons located in different areas of the CNS, such as frontal cortex, brainstem, and spinal cord. And this results in impaired movements, in muscle weakness, paralysis, and eventually this leads to death due to typically respiratory failure within the next 5 years of the initial onset of symptoms. And as these two pathologies, ALS and FTLD, share pathological hallmarks, they are now considered as a single disease entity or a spectrum of disorders, ALS, FTLD. And next, I'll be referring to this disease as ALS, FTLD. So there are two forms of this disorder: familial, 10% of cases, and sporadic, which is up to 95% of cases. And ALS FTLD is caused by a variety of molecular mechanisms, including impaired protein stability, abnormal RNA binding protein function, skeleton damage, impaired DNA repair, and mitochondrial dysfunction. And as we will not dive into all these mechanisms, but as you can see on these pictures, there are several proteins that are involved in many of, or even all of these processes. And these proteins are SOD1, superoxide dismutase 1, TDP43, tRDNA binding protein 43, fused sarcoma, and chromosome 9 open reading frame 72. And mutations in these major established causal ALS FTLD genes account for approximately up to 70% of familial and about 10% of sporadic cases of ALS FTLD. And that is why genetically modified animal models with these ALS causal genes are main models used to study the pathology in rodents. And in our work, we use FUS mutants. These animals have truncated version of this FUS protein. They don't have C-terminal domains that are responsible for nuclear localization of this protein, and this leads to misprocessing of RNAs in the nucleus, and consequently it leads to symptoms of ALS FTLD in these mice. So here on this right picture, you can see a typical FUS transgenic mouse with paralyzed hind limbs. And FUS transgenic mice are characterized by early onset of motor symptoms, 2 to 3 months of postnatal life. And their shorter lifespan, it's around just 4 months. Also, proteinopathy in the CNS causes muscle atrophy. You can see here on the right picture that these mice have much smaller muscles. And here on the bottom pictures, you can see that FUS transgenic mice in their symptomatic stage of disease have neurodegeneration and spinal cord. And what is also interesting is that in all these models of ALS FTLD, in the symptomatic stage of the disease, animals have inflammatory processes going on systemically or in the CNS, neuroinflammation. But for this model, FUS transgenic mice, these inflammatory changes happen even before motor symptom, before developing motor dysfunctions. So in pre-symptomatic mice, we have increased pro-inflammatory cytokines in hippocampus and prefrontal cortex, and also elevated interleukin-1 beta in blood plasma. So it's also a systemic inflammation in these mice. And also pre-symptomatic FUS transgenic mice display several aberrant behaviors, including signs of helplessness, anhedonia. And we hypothesize that systemic infectious inflammation modeled by a single dose of lipopolysaccharide may exacerbate these abnormalities, leading to depressive-like behaviors. So considering that the relationship between FUS-mediated disease and systemic inflammation have not been explored, we sought to investigate emotional abnormalities and inflammatory responses in the CNS during the pre-symptomatic stage of ALS FTLD model by a single dose of LPS to these mice. And in order to look at these changes, we performed 4 experiments. 2 of them were dedicated to more short-term, to look at more short-term changes after LPS injection, and 2 of them were dedicated to look at more distant changes from the LPS injection. And the first part of all 4 experiments is quite the same. We took wild-type mice and food-transgenic mice and injected 1 dose of LPS intraperitoneally and received 4 groups. And in our first experiment, we tested our mice in sucrose preference test, and 24 hours after injection, we looked at the density of activated microglial cells in ventral and dorsal horns of spinal cord, and also in the prefrontal cortex and in hippocampal dentate gyrus. And what we have found is that in sucrose preference test, there is an exacerbation of anhedonia-like behavior in mice that were treated with LPS in food transgenic mice. So they intook much less sucrose solution in comparison to wild-type mice, and also to food transgenic mice that didn't receive LPS. And as for the density of activated microglial cells, we found an increase of this measure in the ventral and dorsal horns of spinal cord induced by LPS in both genotypes. Almost the same we saw in the hippocampus, also increased induced by LPS. But what is interesting, in prefrontal cortex, there was an effect of genotype. So we didn't see an increase of these cells in wild-type mice after LPS treatment, while in food transgenic mice, we did see an increase in this measure. So our next experiment was about testing these animals in oval cage and marble tests. After that, we tested them in forced swim test and looked at the expression of genes of certain pro-inflammatory cytokines in the brain and spinal cord. And in the oval cage, we looked at the number of vertical rearings, and we didn't see any effect of genotype, while LPS induced decrease in this measure in both genotypes in wild type and food transgenic mice. In marble test, there was an effect of genotype. We saw that food transgenic mice displaced much less pellets than wild type mice, and LPS even exacerbated this effect. So food transgenic mice treated with LPS didn't displace any pellets, which tells us about the hippocampal abnormalities in these mice. In the forced swim test, we also saw exacerbation of in forced swim test, we saw an exacerbation of depressive-like behavior evoked by LPS treatment in food transgenic mice, which is indicated by decreased latency to float and increased total duration of floating in these mice. And as for molecular markers of inflammation in the brain and spinal cord, we can see almost the same picture. As for behavioral abnormalities, it's an exacerbation of pro-inflammatory effects of LPS in food transgenic mice for most of these genes that we investigated, interleukin-1 beta in prefrontal cortex, hippocampus, and tumor necrosis factor in PFC, hippocampus, and also in spinal cord. Also, we saw this effect in relation to COX-1 expression in hippocampus and spinal cord, and also in relation to COX-2 expression in prefrontal cortex and spinal cord. And what is also interesting is that Fust transgenic mice had elevated expression of COX-1 in hippocampus even before the LPS treatment. And so to experiments 3 and 4, they were devoted to see more distant changes in behavior and molecular impairments in these mice treated with LPS. So in experiment 3, we investigated how much time it needs for these animals to develop the paralysis. And in experiment 4, we looked at expression of pro-inflammatory cytokines in different regions of CNS 48 hours post-injection, not 24 like in previous experiment, but more distant time point. And we saw no differences between genotypes in these two experiments. So to conclude, we can say that pre-symptomatic Fust transgenic mice have an exaggerated inflammatory response to LPS, and particularly this is evident in prefrontal cortex, as seen by increased microglia activation and elevated cytokine expression. Following LPS injection, both genotypes showed suppression of hippocampus-dependent performance in the marble test and elevated gene expression of interleukin-1 beta, TNF, COX-1 and COX-2 cyclooxygenases in the hippocampus and spinal cord. But the response was always greater in the Fust transgenic mice. Also, these mutant mice exhibited depressive-like behaviors after LPS injection, likely due to increased cytokine expression in the CNS, while no genotype-associated differences in the immune response remained after 48 hours of LPS injection, indicating a rapid decay of the increased pro-inflammatory response in the mutants. And so we can make a general conclusion that this line of mice, Fust transgenic mice, may be used in the study to address the role of microglia inflammation in the mechanisms of ALS FTLD syndrome. And I would like to express my gratitude to all my colleagues and professors who contributed to this work. And I would like to thank you for your attention.
For the presentation, that we could have time only for one question, maybe, if somebody has it, because we're running out of time. Any questions? No questions, thank you. Then we can move to our next speaker. Our next speaker is here. He is present personally, so I think he just finished his lecture. And all questions for discussion can be after that moved to the coffee break zone. Okay. So please, our next speaker is Dr. Evgeny Svirin. He is a researcher to the section of State Medical University of Moscow, and he is guest scientist at Neuroplast company, Moscow, Netherlands.
Саша, экран перестаньте, пожалуйста.
Разверните просто.
Не, нам нужно настроить на потоковое видео.
Да.
Мне кажется, сейчас не будет. Trying to
So we are making the screen looks more pretty when the translator is here, and the demonstration of screen is going from an organizer computer, and that's a bit separate. I can show you after, but since we are not really informatics and IT persons, there are some technical issues that rise up, and we are sorry for them. Just leave it like it was. Just bring it back down.
Просто панелька это куда не денется само по себе. Я не готовы, если видимо, придется трансляции, когда Sorry for the technical issues. So my name is Evgeny Svirin. I work in Sholpan University in Moscow, and I'm also guest scientist at Neuroplast in Netherlands. And I want to share with you some of our findings in APPPS1 mouse model of Alzheimer's disease. We did some behavioral characterization of this model, as well as we tested some compounds which may exert positive effects on Alzheimer's disease. So the Alzheimer's disease is a big problem, actually, because it's a progressive neurodegenerative disorder which affects memory, thinking, and behavior, and it's responsible for 60 to 80% of all dementia cases in the world. And right now, it's estimated that there are 55 million people suffering from Alzheimer's disease. And due to aging of the population, general population, it's expected that this number will be tripled in 30 years. So the problem will be huge. And right now, in high-income countries, it's estimated that Alzheimer's disease is second leading cause of death, actually. And there is no effective cure for now. So the problem is evident. Alzheimer's disease was first described in 1906 by Alois Alzheimer in Germany. He reported case study of a patient, 51-year-old woman, who suffered from memory loss, disorientation, and other cognitive symptoms. As we know now, main symptoms of Alzheimer's disease include memory loss, language disturbances, visual spatial deficits, impaired judgment, motivation, and insight, and also some neuropsychiatric symptoms like depression, anxiety, sleep disturbance, and psychosis. After the patient's death, Alzheimer examined her brain and he found the hallmarks of the disease. So it's a severe deterioration of cerebral cortex and hippocampus due to neuronal death. And and he found the amyloid plaques, not amyloid. He didn't know it's amyloid. He found some plaques and neurofibrillary tangles, which they are cause of neuronal death, which leads to dementia. Here you can see the progression of Alzheimer's disease. It starts in the entorhinal cortex and the hippocampus, which are crucial structures for memory formation. So the memory loss and loss of learning abilities is the first sign, usually first sign of Alzheimer's disease. Then in mild to moderate Alzheimer's disease, we observe increased memory loss and confusion in patients, problems recognizing people, language difficulties, restlessness, agitation, and also repetitive statements. With the further progression of the disease, it's marked with extreme shrinkage of brain. Patient becomes completely dependent on care of other people and suffers from weight loss, seizures, skin infections, increased sleeping, loss of bladder and bowel control, and ultimately it results in death. Right now, there is not much we can do to treat Alzheimer's disease. One promising avenue which we can follow to new treatments is based on the fact that oxidative stress is really important for pathogenesis of Alzheimer's disease. And we can use combinations of the treatments used now with some potential antioxidant compounds, which may be combined for synergistic effect. In the study I will present shortly, we tested several novel antioxidant compounds such as fullerene C60 and its water-soluble form fullerenol, as well as complex of fullerene C60 and plant flavonoid dihydroquercetin, which is synthesized by our colleagues in Portugal and also here in Kazlarda. We used APP-PS1 mice. This mouse model overexpresses two human proteins, which are crucial for Alzheimer's disease. It's amyloid precursor protein and presenilin-1 protein. These mice show really early onset of amyloid plaque formation as early as 6 weeks of age. And they show cognitive and memory impairments. So in this, here you can see in red, these are amyloid plaques in cortex and hippocampus of 5 months old APP-PS1 mice. I will refer to them as just APP mice for shortness. So we took female APP mice and their wild-type litter mates, and from the age of 1 month, we started chronic administration of fullerene C60 with food and fullerenol, which is referred to C60OH, with drinking water. Then at ages of 5, 8.5, and 12 months, we tested these mice in batteries of behavioral tests for emotionality and cognitive behavior. Yeah. Then we euthanized mice and performed brain dissection, and brains were stained for amyloidosis. And we also took blood serum, which I hope will be transferred to Professor Anthony's lab in Oxford for metabolomic assay. So first, the results of memory tests. Here are the results of contextual fear conditioning, which Professor Strekalova told you about. Here we measure the percent of freezing behavior after conditioning with a food shock stimuli. And at age of 5 months, we don't see much difference. Here, this first vehicle group are mice without any treatment, without C6 and without fluororenal. And here, at age of 5 months, they don't demonstrate significant memory loss in this test. But at age of 8.5 months, we already see some significant decrease in freezing behavior in this test, which corresponds to impaired memory formation and learning. Mice which received fluoren and fluorenol actually don't demonstrate such difference in this test. Then at age of 1 year, at 12 months, we tried to perform a step-down avoidance test with the very same mice because we thought that they will forget about this chamber to that point, but that was not the case actually, because wild-type mice still had significantly higher latency to descend from the platform than APP mice. Then, instead of doing the second day of step-down avoidance, during which we usually test the latency to descent, we waited for 2 weeks and then tried to do the memory extinction learning when we placed mice for 20 minutes in the same chamber and they didn't receive food shock there. Then next day, we tried to see the latency, to measure the latency to descent, and we found very interesting effect. In this case, APP mice had actually, yes, we found main genotype effect. So APP mice were actually more reluctant to step down from the platform than wild-type mice. This possibly may be explained that they had delayed consolidation of the memory after this day when they received food shock, and the extinction learning didn't work quite as well for APP mice as it worked for wild types. So this is quite interesting result here. Next, the condition taste aversion test. These are the data with all the compounds we tested, and this looks a little too much, and it isn't yet analyzed statistically. So I'll present data only with vehicle groups. And here we can see that there is a statistical difference between drinking behavior in wild type. I'm sorry, not drinking behavior, sucrose preference in wild type mice. So they actually learned this lesson with lithium chloride, but not in case of APP mice. So memory formation is impaired in 5 months and 8.5 months as well. But interestingly, not at 12 months. Maybe because, first of all, maybe because wild type mice are already old too, and they start to their ability to learn decreases to this age. But also this may be because to this point we already lost some mice, and we just don't have, because of age and because APP mice don't live that well, and the statistical power of this test just diminished to this point. Then in object recognition test, we actually didn't find any memory impairments because there were no statistical differences between object discrimination rates. But we find something that may be referred as to signs of hyperactivity, because at 5 months old, APP mice had greater total time of exploration of objects than wild type mice, and also at age of 8.5 months, they had significantly lower latency to explore new object than wild type mice. Here, here we have some signs of C60 actually working, but this is complex result. I won't go into detail on this right now. Next, we move to tests for emotionality. First of all, open field test. This is quite a versatile test in which we can assess anxiety-like behavior of mice and also general locomotion. Here, we just take like plastic box, put the mice in this box, one mouse at a time, and mice usually naturally avoid open spaces and display so-called stigmataxis, tendency to stick to the borders. And the measure of anxiety-like behavior is time that mice spent in the center of the open field. And here we can see that in case of vehicle group, the amount of time spent in the center is significantly decreased in APP mice. So this is the sign of elevated anxiety-like behavior. Again, this effect, we don't see it in treated groups. Next, two more tests, DAT, light box, and O-maze. Both tests use mice natural aversion to brightly lit, open, and elevated areas, as well as like it was in open field. And here we measure latency and number of so-called risk assessment events. It's just the mice peaks from the dark and closed compounds to the bright one to see what's happening there. And also latency, number, and total time of exits to open or lit compartments. In 8.5 months old APP mice, we see elevated number of risk assessment events, which may be sign of hyperactivity, but also this may be a sign of elevated anxiety. And in this case, we also see a trend towards less time spent in the light box of that light box setup in this mice. So we think that it is anxiety actually rather than hyperactivity. At 1 year old, we see similar trends. And in all cases, these trends are ameliorated by administration of fullerene or fullerenol. Sometimes it seems like fullerene actually has some stimulating effect, because here we can see that there is a significant difference between APP and wild type groups in case of fullerene administration, but it's not because APP spent much more or less, much like, I'm sorry. Well, it's because it tends to affect a wild type group rather than APP. Tube pellet displacement tests or marble test, which was mentioned today already 2 times. Here we also have significant impact of C60 on the behavior, and only slight trends, which can be optical trends, they can be seen here. So APP mice may perform may have decreased performance in this test. And at 1 year old, in the 1 year old mice, we also observed genotype main effect, which means that we have no group differences here, but it seems that APP mice, they tend to perform worse in this test. And this test is hippocampal dependent behavior. So it's expected result for mice who already have lots of amyloid plaques in their hippocampus. We also tested one more compound. It's the complex of C60 and dehydroquercetin. But most of the data from this experiment is still under development, let's say so. And here I want to show you the data from Lyme maze, where we think that there might be a trend towards increased latency to find water reward at days 4 and 5 in APP vehicle mice, but not in APP mice who receive treatment. So maybe this compound works in some way to ameliorate the behavioral symptoms. So the bottom line is that we found signs of increased anxiety-like behavior in 5-month-old APP mice, which persists. And as far as I know, we are first who reported this type of behavior in these mice at such young age. We found, as expected, decreased learning and memory in APP mice at all the ages we measured. And some of the tested compounds, including phytochemical derivative C60DHQ, may be suggested as candidates for future research. Okay, this concludes my talk. Thank you for your attention, and I would like to express my gratitude to everyone who participated in this study.
If you have some questions, I think we have time for 2 questions. Governor, I already you will run out of time. So if any questions, I can.
Could you explain a little bit better the rationale to use of polyren and some other compounds? Also, did you use it as a vehicle, and actually how degradable it is? If you inject it chronically, whether it's going to be excreted or stay in the CNS, it's quite artificial compound.
Yes, to be honest, we are not sure. To be completely honest, we don't know really, because the pharmacodynamics of these compounds, it is not that explored yet. But it seems that fullerene isn't toxic actually at low doses, which we use here. So this is also is left for future research right now.
Do you think it's worth pointing out that what was meant to happen? These are meant to be controls for the soluble fullerene. So bringing together an antioxidant on doing something with C60 in a way that, and then that compound that you finished with wasn't available for a long time. So they started with C60 and it's already given, isn't it? UIV. It's IV or
No, it's oral.
It's oral, yeah. So fundamentally, C60 has to, it's small but big, and it has to cross the gut lumen. It has to get somewhere. So I think what's going to be missing from here that everybody's going to be upset about is this between the gut microbiome or something like that. is somehow, but these are totally inert bloody compounds, so it's not clear to me how it could possibly work. And until we show by mass spec that it's, where it is, so you need to do some, C60 has a really unique mass spec, you'll see it if it's there, and then my suggestion is you look at the gut, you look at the liver, and you look at the brain, and you find out whether any of the C60s left the gut at all. If it hasn't left the gut, then you look to see how you've changed the microbiome in the gut or done something else that makes sense. But right now, I have to tell you, I have no idea why C60 should do, it makes no sense. It would be like, so just that's a bit of a, on the other hand, the other one is meant to be soluble and could easily make its way around the body. So, this, with the antioxidants attached to it.
Is it really soluble in the water? C60OH, fullerenol, yes, it's a bit good.
But the C60 isn't.
C60 isn't, but it is small. So
yeah, but you can see it, can't you? Because they aggregate.
But why is it use oral, not intravenous injection? Because intravenous you would expect better chances to get to the brain when we do orally.
yes, but actually it does aggregate, so intravenous wouldn't be really safe at this point. So if it's not properly emulsified, which I'm not sure how can be done by us. So
it clumps, it forms particles, and I think you should end up with it. I think it'd end up in the lung and it forms a nasty mess. But I do think you have to look for some. You've got an observation now, and you're right. Your other two groups, for whatever reason, look like they're doing better than the control. So, but now you're going to have to find an explanation for it, and it doesn't make a lot of sense. So, my suggestion is the microbiome is a good place to start and find out whether or not you're, conceivably, or I don't know whether it's ever used as a substrate, but it might be somehow changing the way that some of the bugs are behaving in the gut.
Yeah. But when you use two compounds together, do you think it's basically fullerene kind of place where second compound absorbed to the surface? It's like
you mean fullerene dehydroquercetin?
Yeah. Do you think it's kind of we use as a carrier? It's kind of absorbed.
I thought it was carrying in the pound. I thought you managed to find a way of actually peeing it onto the surface.
Yes, it's the chemical structure is like something like C60 dehydroquercetin 4. So one fullerene and 4 makes complex with the 4 molecules of dehydroquercetin. And in this case, it is soluble, water soluble. And then dehydroquercetin is quite a powerful antioxidant itself. So it's like double hit. But right to.
You could go IV with that one, potentially.
Sorry.
Those slides, IV, if you want time, because you can, not, so it's really very.
Oh, it's really can we move to the cost.
Yes.
Thank you for the presentation, and we will thank you for attending to this short day. This is just introduction. Tomorrow we'll have longer, we'll have more speakers tomorrow, and I'm going to really devoted to stem cell therapy as well. So you are welcome to attend. Okay. Thank you. Thank you.
Now we have social meeting.
Now, please, you can proceed to the coffee break room. Welcome.
But so please,
Then the other thing is actually behind the one. I mean,